Hi everybody, welcome back to the urinary system. This is part 2 of our lecture video. Very important that you go back and review the anatomical feature of the nephrons, as we will reference these features, plus the cells, the capillary system, the blood vessels, and of course the tubes that were discussed in the previous chapter. So when we describe urine formation, there are three major processes as outlined here. Step 1 would be glomerular filtration. Step 2, tubular reabsorption. And step 3, tubular secretion. As indicated in this image, you could see where each of these steps is taking place. What's interesting is that the way the kidneys go through the process of urine formation, it's kind of like if someone's going to clean their room, the kidneys are going to remove everything that's inside of the room. So imagine taking out your bed, your drawer, your desks, all of the bulky stuff. Once all that stuff is out, then you sweep everything up, vacuum, and then you put the furniture back in. So that's kind of the process that we're going to outline here. Step 1 is kind of like removing all of the furniture, and you pick up all of those particles that you don't need. And step 2 is kind of like putting back the furniture. And step 3, you're just kind of fine-tuning everything. And so we want to understand that glomerular filtration is really dumping into the waste container. Of course, it takes place in the renal corpuscle and produces cell and protein-free filtrate. We talked about that filtrate is everything that can pass through the pores of the glomerulus, and it enters that bowl, kind of like filtering out your pasta. Tubular reabsorption then is reclaiming what the body needs to keep. So this is the process of selectively moving substances from the filtrate back into the blood. It takes place in the renal tubules and the collecting ducts. Tubular reabsorption reclaims almost everything that's filtered. In other words, tubular reabsorption reclaims back all of the glucose and amino acids, and about 99% of the water, also salt and other components, and anything that's not reabsorbed at this step is going to become urine. Now, tubular secretion, of course, takes place along the length of the tubule and the collecting duct. So this is going to be selectively moving substances from the blood into the filtrate, and your kidneys process an enormous volume of blood each day. Of the approximately 1,200 milliliters of blood that passes through the glomeruli each minute, about 650 milliliters is plasma, and about 1/5 of this, which is about roughly 100 to 125 milliliters, is forced in the glomerular capsules as filtrate. So this is the equivalent of filtering your entire plasma volume more than 60 times a day. So considering the magnitude of their task, it's not surprising that the kidneys, which only account for 1% of our body weight, consume 20 to 25% of all the oxygen used by the body at rest. So filtrate and urine are quite different, as we've been outlining. Filtrate contains everything found in blood plasma except proteins. Urine contains unneeded substances such as excess salts and metabolic waste. The kidneys process 180 liters, which is 47 gallons, of blood-derived fluid daily. And of this amount, 1%, which is about roughly 1.5 liters, typically leaves the body as urine, and the rest is going to return back into the circulation. So let's begin with step 1, which is going to be glomerular filtration. This is a passive process in which hydrostatic pressure forces fluids and solutes through a membrane. The glomeruli can be viewed as a simple mechanical filter because filtrate formation does not directly consume metabolic energy. So we're first going to go ahead and begin talking about the filtration membrane, which is made up of the fenestrated endothelium, the basement membrane, and the foot processes of those podocytes. So remember that the fenestrated endothelium of the glomerular capillaries are going to allow all blood components except blood cells to pass through. And again, it's important to go back and review the anatomy here. The basement membrane is going to be the second part of the filtration membrane. The basement membrane there lies between the other two layers and is composed of their fused basal lamina. It forms a physical barrier that blocks all but the smallest proteins while still permitting some other solutes to pass. Glycoproteins of the gel-like basement membrane give it a negative charge. So as a result, the basement membrane electrically repels many negatively charged macromolecular anions such as plasma proteins, reinforcing the blockade based on molecular size. Let's take a look at the foot processes, which is the third component of the filtration membrane. Remember that the visceral layer of the glomerular capsule is made up of podocytes that have filtration slits between their foot processes. So molecules that are smaller than 3 nanometers in diameter, such as water, glucose, amino acids, and nitrogenous waste, can pass freely from the blood into the glomerular capsule. So as a result, these substances usually have similar concentrations in the blood and the glomerular filtrate. Larger molecules pass with great difficulty, and those that are larger than 5 nanometers are generally inhibited or they're barred from entering the tubule. Keeping the plasma proteins in the capillaries maintains what we call a colloid osmotic pressure of the glomerular blood, preventing the loss of all its water to the capsular space. And some of you know that the presence of proteins or blood cells in the urine usually indicates a problem with the filtration membrane. Now, the principles that govern filtration from the glomerulus are the same as those that govern filtration from any capillary bed. So if we take a look at this image, we see that these principles are going to be applied to the glomerular capillaries of the nephron. So there are outward pressures that we need to outline. The first one is going to be the hydrostatic pressure in the glomerular capillaries, and we abbreviate this as HPGC. And this is essentially glomerular blood pressure. It is the chief force pushing water and solutes out of the blood and across the filtration membrane. The blood pressure in the glomerulus is extraordinarily high. So it's about 55 millimeters of mercury compared to an average of most capillary beds, which is about 26 millimeters of mercury. And it remains high across the entire capillary bed. So this is because the glomerular capillaries are drained by high resistance efferent arterial, whose diameter is smaller than the afferent arterial that feeds them, as we outlined in the previous video. So as a result, filtration occurs along the entire length of each glomerular capillary, and reabsorption does not occur as it would in other capillary beds. So now that we've discussed the outward pressure, let's move over to the inward pressures. So there are two that we need to discuss. The first one's going to be the hydrostatic pressure in the capsular space, and this one's abbreviated HPCS, and this is the pressure exerted by filtrate in the glomerular capsule. The hydrostatic capsular space is much higher than hydrostatic pressure surrounding most capillaries, just because filtrate is confined in a small space with a narrow outlet. The colloid osmotic pressure is the other one that we need to go ahead and discuss. And this is colloid osmotic pressure in glomerular capillaries, which is abbreviated OPGC. So this is the pressure exerted by the proteins in the blood. So as we look at this image one more time, the above pressure represents the net filtration pressure, or NFP, which largely determines the glomerular filtration rate, which is what we're going to consider next. So if we think about the glomerular filtration rate, or GFR, is the volume of filtrate formed each minute by the combined activity of all 2 million glomeruli of the kidneys. GFR, therefore, is directly proportional to each of the following factors. It's proportional to the net filtration pressure, which is the one that we saw here, the 10 millimeters of mercury. So NFP is the main controllable factor. Of the pressures determining NFP, the most important is the hydrostatic pressures in the glomerulus. This pressure can be controlled by changing the diameter of the afferent arterioles, as well as we're going to outline shortly. The next component that is going to go ahead and directly affect GFR is going to be the total surface area available for filtration. So glomerular capillaries have a huge surface area. Collectively, the surface area is equal to the surface area of the skin. Glomerular mesangial cells surrounding these capillaries can fine-tune GFR by contracting to adjust the total surface area available for filtration. And then the last component of GFR is going to be the filtration membrane permeability. So glomerular capillaries are thousands of times more permeable than other capillaries because of their fenestrations. Therefore, all of these three combined are going to be important in that they are directly going to control glomerular filtration rate. Finally, of the 180 liters of filtrate that is produced daily by the kidneys, that 180 liters filtrate per day translates to the normal GFR, which is going to be 120 to 125 milliliters per minute. So another thing to think about is that assuming nothing else changes, an increase in the GFR is going to increase urine output, which reduces blood volume and blood pressure, and the opposite holds true for a decrease in GFR. There are two types of controls that your body uses in order to make sure that this glomerular filtration rate stays at the normal 120 to 125 milliliters per minute. So we're going to talk about intrinsic controls, which are going to be renal autoregulatory, and they act locally within the kidney to maintain GFR, while the extrinsic controls are by the nervous system and endocrine systems to maintain blood pressure. In extreme changes of blood pressure, meaning that the mean or MAP, mean arterial pressure, less than 80 or greater than 180 millimeters of mercury, extrinsic controls take precedence over intrinsic controls in an effort to prevent damage to the brain and other crucial organs. GFR can be controlled by changing a single variable, and that single variable is glomerular hydrostatic pressure. All major control mechanisms act primarily to change this one variable. If the glomerular hydrostatic pressure rises, so does the NFP, and so does GFR. If the glomerular hydrostatic pressure falls by as little as 18%, GFR can drop to 0. Clearly, hydrostatic pressure in the glomerulus must be tightly controlled. So let's take a look at how the intrinsic mechanisms are going to work. Intrinsic controls are going to be maintained or work as long as the mean arterial pressure is between the 80 to 180 millimeters of mercury. There are two types of renal autoregulation that we need to discuss. The first one is called myogenic mechanism, and the second is called tubular glomerular feedback mechanism. So with renal autoregulation, the kidney can maintain nearly a constant GFR despite fluctuations in systemic arterial blood pressure. The myogenic mechanism, which is the first one that we were going to cover, reflects a property of vascular smooth muscle. It contracts when stretched and relaxes when not stretched. So rising systemic blood pressure stretches vascular smooth muscle in the arteriolar walls, causing the afferent arterioles to constrict. So this constriction restricts blood flow into the glomerulus and prevents glomerular blood pressure from rising to damaging levels. Declining systemic blood pressure causes dilation of afferent arterioles and raises glomerular hydrostatic pressures. So both responses help maintain the normal NFP and consequently the normal GFR. So this contraction and dilation, hence, is the reason why we refer to this first intrinsic control myogenic mechanism. The second mechanism is the tubular glomerular feedback mechanism, and it's essentially directed by the macula densa cells, or the juxtaglomerular complex that we learned about in the previous video. So these cells, remember, are located in the walls of the ascending limb of the nephron loop, and they respond to filtrate, sodium chloride concentration, which varies directly with filtrate flow rate. When GFR increases, there is not enough time for reabsorption, and the concentration of sodium chloride in the filtrate is going to remain high. The macula densa cells respond to high levels of sodium chloride in filtrate by releasing vasoconstrictor chemicals, some of which are ATP, and these chemicals cause intense constriction of the afferent arteriole, reducing blood flow into the glomerulus. This drop in blood flow decreases NFP and, of course, GFR, slowing the flow of filtrate and allowing more time for filtrate processing and also that sodium chloride reabsorption. So in contrast, the low sodium chloride concentration of slowly flowing filtrate inhibits ATP release from macula densa cells, causing vasodilation to the afferent arterioles. So this allows more blood to flow into the glomerulus, thus increasing NFP and ultimately the GFR. Autoregulatory mechanisms maintain a relatively constant GFR over an arterial pressure range that we just discussed earlier. The range has to be between 80 and 100 millimeters of mercury. So consequently, normal day-to-day changes in our blood pressure, such as when we exercise or we sleep, do not cause large changes in water and solute excretion. However, the intrinsic controls that we just discussed, these two, myogenic and tubular glomerular feedback, they cannot handle extremely low systemic blood pressure, such as might result from a serious hemorrhaging or hypovolemic shock. Once the mean arterial pressure drops below 80 millimeters of mercury, the autoregulation stops or ceases, and the extrinsic controls are going to take over. So again, the purpose of the extrinsic controls regulating the GFR is to maintain systemic blood pressure. So when the volume of the extracellular fluid is normal and the sympathetic nervous system is at rest, the renal blood vessels are dilated and the renal autoregulation mechanism prevails, as we previously outlined. However, when the external or extracellular fluid volume is extremely low, as in hypovolemic shock during severe hemorrhaging, it is necessary to shunt blood to vital organs, and neural controls may override autoregulatory mechanisms. This could reduce renal blood flow to the point of damaging the kidneys. So when blood pressure falls, norepinephrine, which you know is released by sympathetic nerve fibers, and epinephrine from the adrenal medulla, causes vascular smooth muscles to constrict. So thereby, this increases peripheral resistance, bringing blood pressure back up toward normal. This is the baroreceptor reflex we discussed back in the chapter with the blood vessels. So as part of these reflex, the afferent arteriole also constricts. Constriction of the afferent arterioles decreases GFR and so helps restore blood volume and blood pressure to normal. So that's going to be the sympathetic nervous system. The second extrinsic control is one that you're very well familiar with, hopefully by this time, as we discussed and introduced it first in the endocrine system. We also talked about this when we were dealing with the heart and blood vessels. We know this as the RAS, the renin-angiotensin-aldosterone mechanism. So this is the body's main mechanism for increasing blood pressure. Without adequate blood pressure, again, as we just talked about hemorrhage or dehydration, glomerular filtration is not possible, so this mechanism regulates GFR indirectly. Low blood pressure causes the granular cells of the juxtaglomerular complex to release renin. And there are three pathways that can stimulate granular cells. So the first one is sympathetic nervous system. So as the baroreceptor, or as part of the baroreceptor reflex, renal sympathetic nerves activate the androgenic receptors that cause the granular cells to release renin. Secondly, you can also have activated macula densa cells. So low blood pressure or vasoconstriction of the afferent arterioles by the sympathetic nervous system reduces GFR, slowing down the flow of filtrate through the renal tubules. So when the macula And then finally, you have reduced stretching. So granular cells can act as mechanoreceptors. A drop in mean arterial blood pressure reduces the tension in the granular cells' plasma membranes and stimulate them to release more renin. So these would be the three pathways that are going to act on renin release. So let's take a look at this image, which shows us or reviews the intrinsic and the extrinsic controls that we just discussed. So we mentioned that intrinsic controls are going to directly regulate GFR despite moderate changes in blood pressure when the MAP is between 80 and 100 millimeters of mercury. So if we have a drop in blood pressure in the afferent arterioles, this of course causes a drop in GFR. So as a result, you have the stretching. These are myogenic mechanisms, stretching of smooth muscles in walls of the afferent arterioles, which is going to vasodilate, and this is going to cause an increase in GFR. And then here you have the tubular glomerular mechanisms of autoregulation. So these again are all the intrinsic by the kidneys. The extrinsic mechanisms again are going to indirectly regulate GFR by maintaining a systemic blood pressure, which drives filtration in the kidneys. We looked at the act of the nervous system, and then we also looked at the role that the granular cells play in releasing renin, and we saw previously that there are three ways that these cells can be stimulated to release renin. There is a video on Canvas that you're going to find that explains to you the concept of the RAS system, which we outlined in the endocrine system, and also where we're learning the cardiovascular system. And this is going to increase systemic blood pressure, which indirectly is going to regulate GFR. So you can study this image a little bit more closely. So that is going to be our step 1, which is glomerular filtration of our pathway through the urine formation that we want to outline. Now we're going to move over to step 2, which is going to be tubular reabsorption. And we're going to learn that this is actually a selective process, where we learned that step 1, glomerular filtration, was a passive process. So our total plasma volume filters into the renal tubules about every 22 minutes. So all our plasma would drain away as urine really in less than 30 minutes, were it not for this step, tubular reabsorption, which quickly reclaims most of the tubule contents and returns them to the blood. Tubular reabsorption is selective, and it's conducted by transepithelial process, and it begins as soon as the filtrate enters the proximal tubules. So to reach the blood, reabsorbed substances have to follow either a transcellular or a paracellular route, as you're seeing in this image. In the transcellular route, what happens is that the transported substances move through the apical membrane, the cytosol and the basolateral membrane of the tubule cells, and then the endothelium of the peritubular capillaries. The movement of substances in the paracellular route happens between the tubule cells, and this is limited by the tight junction connecting these cells. In the proximal nephron, however, these tight junctions are leaky and allow water and some important ions such as calcium, magnesium, potassium, and some sodium as well to pass through the paracellular route. So if you study this image, you see that the transcellular route is going to transport across the apical membrane, diffusion through the cytosol, and then you have transport across the basolateral membrane, and then movement through the interstitial fluid and into the capillaries. So these would be all of the water, and some of the solutes that we just mentioned are going to leave the filtrate, i.e. in the tubules, and we want all this stuff to go back into the blood supply. So this would be the peritubular capillaries that surround the tubes, as we outlined in the previous lecture. And the paracellular route thereby is movement through leaky tight junctions, particularly in the proximal convoluted tubule, as you see here. It's more of a direct movement, if you will. So let's take a look now at the difference between the active tubular reabsorption and the passive tubular reabsorption. So if we think about a healthy kidney, virtually all organic nutrients such as glucose, amino acids, are completely reabsorbed to maintain or restore normal plasma concentrations. So on the other hand, the reabsorption of water and many ions is continuously regulated and adjusted in response to hormonal signals. So depending on the substance transported, the reabsorption process may be active or passive. When we think about active tubular reabsorption, we're going to note that this actually requires ATP, either directly or indirectly. And passive tubular reabsorption encompasses diffusion, facilitated diffusion, and osmosis. And a lot of these substances, of course, when you're thinking about passive, move down their concentration gradients, which you probably remember from your first A&P class. Here is a very good table helping you understand what are these substances that are going to be reabsorbed, what is the main mechanism of reabsorption, and where they're being reabsorbed. So let's take a look at, for example, sodium. So sodium is going to be primarily actively transported via the vaso or basolateral sodium-potassium pump, as you recall, and you have also through these channel proteins, symporters and antiporters. The nutrients such as glucose, amino acids, vitamins, they're going to be transported via secondary active transport with the sodium. When you're dealing with chloride, potassium, calcium, these are going to be passive, paracellular diffusion. Water is by osmosis, of course. Lipid solubles, passive diffusion driven by concentration gradients. And urea is primarily passive, paracellular diffusion. And again, these are going to be happening in the proximal convoluted tubule. When we look at the nephron loop, the descending and the ascending, water is going to be reabsorbed by the descending limb, and the ascending limb is pretty much going to be taking care of these ions. So that will be the ascending limb. And again, you could see that these ions, secondary active transport, calcium and magnesium, passive paracellular diffusion. Let's move over to the DCT again. So you have sodium and chloride and the mechanism. So this is a good table to have on hand when you get a quiz question or an exam question. You can always reference this table. So here would be the written component that we just described. So the PCT, one thing to keep in mind is that this is the site of most reabsorption. So essentially all nutrients such as glucose and amino acids are being reabsorbed. 65% of the sodium and water are being reabsorbed. A lot of those ions that we just looked at previously are being reabsorbed. All the uric acid is going to be reabsorbed. Half of urea, and we'll see why some of that urea has to be secreted back into filtrate, because you want those components that you don't need in the blood, of course, to become urine or be excreted out. In the nephron loop, or in the loop of Henle, we see that the descending limb, as we outlined in the previous lecture, because of the permeability of the cells that make up the descending limb, water can leave. So the descending limb, we want to make a point that water is actually, it's permeable to water. Water is able to move in and out by osmosis. And here's the rule with the descending limb. If water can leave, solutes cannot. And the opposite is true with the ascending limb. In other words, the ascending limb is impermeable to water, but solutes can leave. Why is this? we'll talk about how this is important with the countercurrent exchange system. It has to do with making sure we fine-tune and we regulate the amount of water that needs to be reabsorbed back into the blood in order to work with the blood volume, and likewise the amount of salt that needs to either stay in the blood or leave the body. Let's look at the distal convoluted tubule and the collecting duct. In the proximal convoluted tubule, a nephron loop does not vary with the body's needs. Hormones fine-tune reabsorption in the distal convoluted tubule and also the collecting duct. So because most of the filtered water and solutes have been reabsorbed by the time the distal convoluted tubule is reached, only a small amount of filtered load is subject to its fine-tuning. So about 10% of the original filtered sodium chloride and 25% of the water. What's important to note here is that the reabsorption process in the DCT and collecting duct is reabsorbed and hormonally regulated. You remember these hormones: antidiuretic hormone, aldosterone, ANP, atrial natriuretic peptide, and also PTH, parathyroid hormone. So the ADH, as we've learned in the endocrine system, ADH is going to inhibit diuresis or inhibits urine output. And the way it does that is that the amount of ADH determines the number of aquaporins and thus the amount of water that's reabsorbed. When the body is overhydrated, extracellular fluid osmolality decreases, hence decreasing ADH secretion by the posterior pituitary and making the collecting duct selectively impermeable to water. ADH also increases urea reabsorption by the collecting ducts, as we will describe a little later. Aldosterone is going to fine-tune the reabsorption of the remaining sodium. We have mentioned that when sodium is reabsorbed, water is going to follow. This is going to be important because decreased blood volume or blood pressure or high extracellular potassium concentration, also known as hyperkalemia, can cause the adrenal cortex to release aldosterone to the blood. So except for hyperkalemia, these conditions promote the renin-angiotensin-aldosterone mechanism that we have highlighted previously. Aldosterone targets the principal cells of the collecting ducts and cells of the distal portion of the DCT. As a result, little or no sodium leaves the body in urine. In the absence of aldosterone, these segments reabsorb much less sodium, and about 2% of the sodium filtered daily can be lost, an amount incompatible with life, of course. So physiologically, aldosterone's role is to increase blood volume and therefore blood pressure by enhancing sodium reabsorption. And as we mentioned, water generally follows sodium in aquaporins if the aquaporins are present. Aldosterone also reduces blood potassium concentrations because aldosterone-induced reabsorption of sodium is coupled by potassium secretion in the principal cells of the collecting duct. So that is that sodium enters the cell and potassium moves into the lumen. The next hormone that you may remember is atrial natriuretic peptide, also known as ANP. So in contrast to aldosterone, which acts to conserve sodium, ANP reduces blood sodium, thereby decreasing blood volume and blood pressure. So released cardiac atrial cells when blood volume or blood pressure is elevated. ANP exerts several effects that lower blood sodium content, including direct inhibition of sodium reabsorption of the collecting ducts. And then the last hormone is PTH, which is acting primarily on the distal convoluted tubule. PTH increases the reabsorption of calcium. So the next part of the video, part 3, we will discuss the last step in urine formation, which is tubular secretion, and we will also talk about the countercurrent system, which is going to regulate urine concentration and volume.